Hello friends, welcome to my course on microbial biotechnology. We are in module number 6 discussing industrial and pharmaceutical applications of microorganisms. Today we will be discussing about microbial enzymes of industrial importance. The lecture is divided into 3 broad sections. The first section we try to give an introduction, we will give an overview, then the enzyme classification and nomenclature, then enzymatic synthesis versus chemical synthesis, and we will discuss about the advantages of microbial enzymes. The section 2 we will be discussing about the production of microbial enzymes. In section 3 we discuss the strategies to improve microbial enzymes, and in section 4 we will be discussing about the applications of microbial enzymes in various industries. So, let us first have an overview of microbial enzymes in industry. Microbial enzymes are essential metabolic catalysts driving over 500 industrial products and meeting the growing demands for sustainable solutions. Produced either extracellularly or intracellularly by various microorganisms such as bacteria, actinomycetes, fungi, and these enzymes exhibit diverse structures and serve a wide range of commercial applications. Of about 4,000 known microbial enzymes, around 200 types are used commercially, but only about 20 are produced on a truly industrial scale. These enzymes support approximately 150 industrial processes, including applications in pharmaceuticals, food production, and environmental management. The global industrial enzyme market is highly competitive and led by major players such as Novozymes, DSM, and DuPont. While North America and Europe are currently the largest consumers, the Asia Pacific region, particularly China, Japan, and India, is witnessing rapid growth in enzyme demand due to expanding economies and industrial advancements. Let us look into the various microbial enzymes that are used in numerous industries. It is used in the field of microbiology, molecular biology, for example, like DNA ligases, restriction enzymes, polymerases, which are mostly DNA manipulative or DNA modifying enzymes. Then enzymes of microbial origin have applications in food and dairy industry like proteases, lipases, lactase, pectinase, amylase, lactase, amyloglucosidase, phospholipase. Then in the pharmaceutical and therapeutics also it has wide applications like example penicillin acylase, peroxidase. Then it has applications in the paper and pulp industries where amylase, lipase, protease, cellulase, hemicellulase, esterase, ligninase and xylanase are used. Then these enzymes are used in the organic synthesis industry like lipases, oxidoreductases, glycosyltransferases, lyases, and then it is used also in the detergent industry like example amylase, cellulase, lipase, protease, manage. Then these are used in the waste treatment, for examples amylases, cellulases, lipases, actinases, proteases, lactases and oxygenases, and these are also used for the production of animal feed like phytase and xylanase, and it is widely used in ethanol production like cellulases, ligninases and manganase. Then it is used in the leather processing industry like proteases and lipases, and also in the textile industries like amylases, cellulases, pectinases, catalases, proteases, peroxidases and keratinases. So, we see that some of these enzymes are common to many of these application fields and some are specific to the particular fields. Now, let us try to learn about the enzyme classification of the International Union of Biochemistry and Molecular Biology, shortly known as IUB-MB, which classifies over 3,000 identified enzymes into 7 groups designated as enzyme classes EC 1 to 7 based on their catalytic functions. For example, EC 1 there are the oxidoreductases. These catalyze oxidation reduction reactions. Examples are reductases, oxidases and dehydrogenases. Then EC 2 transferases transfer functional groups between molecules, example transaminases, kinases and glycosyl transferases. Then we have the EC3 hydrolases, use water to break bonds in large molecules, example proteases, amylases and lipases. So, then there is the EC4 lyases. This catalyze addition elimination reactions without hydrolysis of oxidation, Examples are decarboxylases, dehydratases. Then we have the EC5 isomerases. This facilitate intramolecular rearrangements, example racemases and epimerases. Then we have the EC6 ligases, they join molecules using ATP hydrolysis, example synthesis, DNA ligases, carboxylases. Then we have EC7, the translocases, transport ions or molecules across membranes, example ADP, ATP translocases. In this figure you can see the generic working principle of translocases. The enzyme nomenclature system is a standardized method for naming and classifying enzymes established by the International Union of Biochemistry and Molecular Biology. This system classifies enzymes based on the type of reaction they catalyze and gives a unique identification number to each enzyme, ensuring uniformity and consistency in enzyme naming. Each enzyme is assigned a unique numerical code which consists of 4 parts, example EC 2.7.1.1 which stands for hexokinase. The first digit designates the main class, specifies one of the 7 main classes of the enzymes. The second digit indicates the type of group or bond acted upon or the subclass. So, third digit is subclass, describes the reaction type of further categorizes the group affected. The fourth digit uniquely identifies the specific enzyme within the subclass. Now, let us have a comparative discussion between the enzymatic synthesis and chemical synthesis. The chemical synthesis suffers from various disadvantages, like it requires high temperature, low pH and high pressure. It has low catalytic efficiency. It lacks enantiomeric specificity, and it uses organic solvents, which leads to organic waste and environmental pollutants. On the other side, enzymatic synthesis has a lot of advantages. For example, as against the high temperature required by chemical synthesis, these can operate efficiently under moderate temperatures, neutral pH and atmospheric pressure. And as against the low catalytic efficiency of chemical synthesis, enzymatic synthesis offers better efficiency and it has long half life. Similarly, advantages of enzymatic synthesis provides high stereoselectivity, resulting in both stereochemically defined products and radiochemically defined reactions. Enzymatic synthesis, as against the pollution caused by chemical synthesis, is eco-friendly. So, we see that today in this age of climate change and sustainable development, enzymatic synthesis is going to play a very crucial role. So, what are the advantages of microbial enzymes over other sources? Microbial enzymes account for 85% of the industrial enzyme production, with fungi and yeast contributing 50%, bacteria 35% and plants the remaining 15%. Compared to plant and animal enzymes, microbial enzymes offer several advantages, like it has higher activity and stability. Microbial enzymes are more active and stable, making them suitable for large scale fermentation processes using selected strains. It is also very easy to produce the microbial enzymes and also optimize the production process. They yield high quantities and can be easily modified and optimized due to their biochemical diversity and susceptible to genetic manipulation. The microbial world offer rich source of discovery. The modern techniques such as metagenome screening, genome mining and exploration of extremophiles enables the discovery of novel microbial enzymes with unique properties. Now, let us go to the next section where we will discuss about the production of microbial enzymes. We start with a general outline of manufacturing enzymes. Then we go on to discuss about the enzyme discovery by the metagenomic screening and enzymes from extremophiles. We also discuss about enzyme inhibitors and fermentation conditions, the use of inexpensive substrates to cut down cost and optimization of the fermentation process. And once the enzyme is produced by the microbe, how we purify those enzymes. The production of industrial enzymes from microorganisms involves several key steps like isolating and identifying enzyme producing microbes, optimizing production conditions, fermentation, enzyme purification and final formulation for commercial use. Most industrial enzymes are produced using genetically modified bacteria and fungi to enhance yield. Two primary fermentation methods, solid state and submerged fermentation, are used, with submerged fermentation being preferred due to the extracellular release of enzymes into the production medium. Factors like pH, temperature stability, specificity and response to activators and inhibitors influence enzyme selection. Bacillus and Aspergillus species are among the most commonly used microorganisms for industrial enzyme production. Let us discuss about the general outline of enzyme manufacturing. So, we have basically two important operations. One is the upstream set of operations and other is the downstream processing operations. So, in the upstream processing set of operations, we start with the isolation and screening of microorganisms which will be producing these industrial enzymes. Then we go for preparation of the microbial inoculum, then production of enzymes under optimized fermentation conditions. Once the enzymes is produced, we go for the downstream processing operations. It may be of different kinds. We first centrifuge to get the crude enzyme and then also purify the enzyme further, and then we also characterize the enzyme or go for quality control check. Now, let us discuss a little bit about discovery of novel enzymes through the metagenomic screening approach. So, there are countless microbes in the biosphere, but only 1% of them can be cultivated using the known laboratory techniques, which we refer to as the standard laboratory techniques. To overcome this limitation, metagenomics has emerged as an alternative approach, allowing the construction of genomic libraries from environmental DNA. These libraries are systematically screened for open reading frames that encode potential novel enzymes. Studies from diverse habitats such as volcanic vents, Arctic tundra, cow rumen, marine environments and termite guts have identified microbial enzymes with promising biocatalytic applications. So, here you can see some termites and a cow, and onto which a diagram of its rumen is superimposed. We use metagenomic advancements in isolating microorganisms and screening the enzymes from such complex environments. Then another approach is the sequence based screening of metagenomic libraries. Sequence based screening involves detecting genes homologous to known sequences using computational and molecular techniques. Recent advances in genome sequences have led to a wealth of data in sequence databases, creating opportunities for discovering new natural products including enzymes through database mining. Currently, over 2000 genome sequences and draft assemblies are available in the NCBI database. These genome sequences are exploited for discovery of new enzymes using any of the following two strategies. The first strategy is the genome hunting. This approach involves searching for open reading frames or ORFs in a genome specific microorganisms. Sequences annotated as putative enzymes are then cloned, overexpressed and screened for activity. So, we do not clone the enzymes, we clone the gene sequence that produce these putative enzymes. So, the next is the data mining. This method uses bioinformatics tools like BLAST to search for conserved regions across all sequences in databases, identifying homologous protein sequences that may be candidates for further characterization. Function based screening of metagenomic libraries. So, a function based screening of metagenomic libraries is used for detection of hits by introducing foreign genes in expression systems such as Escherichia coli, Streptomyces lividans, Pseudomonas putida and Rhizobium leguminosarum among others. The hit rate in function based metagenomics or the probability of identifying a target gene depends on factors such as the gene size and the sensitivity of the assay method used. So, here we have the environmental sample from where we obtain the heterologous genomic DNA, and then we clone them into certain vectors and then transform them into host cells. Then we go for two kinds of approaches. One is the function driven analysis, and another is the sequence driven analysis using these common metagenomic library. The enzyme activities are typically screened on agar plates supplemented with specific substrates. Now, a question may arise why we need to go for cloning. So, I need to re-emphasize once again that only 1% of the known bacterial diversity species are culturable, remaining 99% are non-culturable. So, these DNA basically is targeted towards the non-culturable organisms. So, these are obtained and then these are cloned and in vectors and transformed into bacterial strains which are culturable. So, by these method we are able to generate a metagenomic library in a host bacteria which is culturable, so that the hidden useful genes in the non-culturable microbial population are being able to be expressed and studied or analyzed. Let us now discuss about the discovery of enzymes from extremophiles. As a term, extremophiles is now already known to you. These are organisms which can survive in extreme conditions, or they love extreme conditions due to their ability to thrive in harsh environments with the extreme conditions such as temperature from very low to very high, -2 to 12 degree and then 60 to 110 degree centigrade, and also high pressure, then in the presence of radiation and under salinity, highly saline conditions like 2 to 5 NaCl, and in extremely low pH as low as 2 or high pH as high as 9. And these extremophiles are valuable sources of enzymes that exhibit exceptional stability under conditions considered incompatible with biological materials. Recent studies suggest that the diversity of organisms in these extreme environments may be even greater than what was thought previously. However, since most of these microorganisms are yet to be isolated in pure culture, characterizing their enzymes remains a significant challenge. Here in this picture you can see Thermus aquaticus deposited on a millipore filter, and we know the importance of Thermus aquaticus which provides us the Taq polymerase, about which we will discuss at length. Enzymes from thermophiles and hyperthermophiles like proteases, lipases, cellulases and amylases are increasingly utilized in various industrial applications. Thermophiles which can thrive at temperatures between 60 to 80 degrees centigrade or even up to 110 degrees centigrades include members of the bacterial genera like Clostridium thermus, Thermotoga and Bacillus, while hyperthermophiles which endure even higher temperatures belong mainly to archaea such as Pyrococcus, Thermococcus and Methanopyrus. The Taq DNA polymerase from Thermus aquaticus alone generated $500 million in sales in 2009. Let us now discuss about enzymes from psychrophiles. Enzymes from psychrophiles or coal-loving organisms are also gaining attention due to their potential for reducing energy consumption in industrial processes. In this picture is shown Psychrobacter phenyl pyrovicus, which is a psychrophile on Columbia horse blood agar. These coal-active enzymes like for example proteases, amylases and lipases are ideal for applications such as in detergent formulations, which help reduce textile wear as well as polymer degrading activities, example cellulases, gyllanases used in the pulp and paper industry and the production of second generation biofuels from lignocellulosic biomass. These enzymes also find applications in fruit juice extraction, bakery improvements, textile polishing and bioremediation of oil contaminated water. Let us now discuss about enzymes from halophiles. Halophiles thrive in high salt concentrations and they produce enzymes which are adapted to these high salt conditions. Some examples include gyllanases, amylases, proteases and lipases. These enzymes derived from such halophiles have been used in non-aqueous media and those from genera like Halobacterium, Halobacillus and Halothermotrix shows great potential for industrial use. There are also enzymes which have been isolated from alkaliphiles. These are microorganisms that can tolerate extreme pH, offer a valuable source of thermoalkaliphilic enzymes like proteases and lipases which are useful as additives in laundry and dishwashing detergents. Now, let us focus a little bit on the various fermentation conditions. Industrial enzymes are produced through carefully controlled fermentation processes using microorganisms, some of which were discussed prior to these, ensuring consistent quality, optimal yields and the ability to meet industrial demands. Two primary methods are employed: submerged fermentation and solid state fermentation. Here in this picture you can see the submerged fermentation. Here microorganisms grow in a liquid nutrient medium, enabling efficient production of extracellular enzymes which are secreted directly into the medium in large quantities. This facilitates easier enzyme recovery and purification, reducing production costs and improving efficiency. Additionally, submerged fermentation allows precise control of culture parameters such as pH, temperature, dissolved oxygen and nutrient supply, ensuring optimal growth conditions, maximum yields and product consistency. Another type of fermentation is the solid state fermentation. The limitation of SMF is its reliance on costly synthetic media. In contrast, solid state fermentation uses minimal liquid compared to SMF and offers a theoretically simpler and more cost effective downstream processing. Solid state fermentation has several key advantages such as high volumetric productivity which allows efficient enzyme production within a smaller fermentation volume, higher product concentration which reduces the need for extensive purification and reduced effluent generation, making it more environmentally and economically favorable. Utilizing inexpensive byproducts or agricultural residues as fermentation substrate not only lowers production cost, but also mitigates environmental pollution caused by their disposal. Selecting cost effective substrates can significantly reduce process costs, potentially cutting them by 1/3. For example, Bacillus megatherium produced substantial amounts of keratinase using chicken feeders as both carbon and nitrogen sources. So, here we can see two commonly used agricultural residues used for enzyme production, the coconut cake on the top and wheat bran below. Other agricultural residues have been employed for enzyme production by various Aspergillus species including phytase, keratinase and lacase, and coconut cake has been used for cellulose secretion by Pseudomonas fluorescens. In this table we can find many examples of enzymes produced from inexpensive substrates. Basically the substrate is used to grow the microorganism which produces these enzymes. So, some of the substrates include, apart from the chicken feeders, jamun leaves, molasses, orange peel moistened with molasses. Then we have the organic kitchen wastes, rice bran and wheat bran, sawdust, sugarcane bagasse or straw, wheat bran, dry corn sawdust, tea stalks and wheat bran. And then The microorganisms which are grown on these include, for example, in chicken feeders we can grow Bacillus megatherium. Aspergillus species can be grown in jamun leaves, molasses, orange peel moistened with molasses, and even sugarcane bagasse. And then even tea stalks, and also in wheat bran. Then we can grow Bacillus lehensis in rice bran and Penicillium on the sawdust. And you can see the various enzymes produced by these organisms, which are listed in the central column. So, the production of industrial enzymes by microorganism is heavily influenced by factors such as incubation time, sacking agitation, initial pH, inoculum concentration, incubation temperature, carbon source, metal ions, and nitrogen sources. So, it is very important to optimize the fermentation conditions. Optimizing these factors is crucial for maximizing the enzyme yields. Optimization involves fine tuning media components, cultural parameters, and fermentation conditions. Typically, this is done using a one factor at a time approach, where a single variable is optimized while keeping all others constant. The optimized condition is then applied sequentially in subsequent experiments. This systematic optimization of nutritional, physicochemical, and fermentation parameters is essential for developing a cost effective and efficient fermentation process, enabling the production of industrial enzymes in adequate quantities. Once the enzymes are produced by any of those fermentation methods we have discussed and growing them on the various substrates that we have discussed, we need to purify, isolate the enzymes. So, the commercial enzymes are produced in bioreactors and are initially obtained in crude form, requiring extraction and purification for further applications. These microbial enzymes are typically purified using three major approaches. Number one is based on ionic properties of the enzyme. The second is based on the ability to get absorbed, and third is based on differences in the size of molecules. So, here in the first case we can see the ion exchange chromatography, where particles with charges opposite to the bead bind to it, and this principle is used for the purification. Then we see in the second the size exclusion chromatography. Here smaller particles traverse slower as they enter the pores on the beads and thus have more distance to cover, whereas the larger particles which do not enter these beads will travel faster. Then we have the adsorption chromatography, where particles with affinity for the beads are adsorbed onto the beads, while the others which do not have that affinity move away quickly. So, exploiting these various properties based on various principles, we can purify the enzymes. Industrial scale chromatography is widely used for enzyme purification, with key applications including desalination of enzyme solutions and batch preparations. Advances in the stability and hydraulic properties of chromatographic media have made these techniques suitable for large scale production. Critical factors for scaling up chromatographic systems include the column height, which ensures efficient separation and resolution, then the linear flow rate, which maintains optimal flow to balance speed and separation efficiency. Then the sample volume to bed volume ratio, this prevents overloading the column and ensures effective purification. In this photo you can see an industrial scale supercritical fluid chromatographic systems. We now move on to the next section, where we will be discussing about strategies to improve microbial enzymes. Here we will discuss about the rational design, site directed mutagenesis and site directed saturation mutagenesis. Then we also discuss about directed evolution, then expression of recombinant enzymes, truncation and fusion. The increasing use of enzymes across various industries is driving a demand for biocatalysts with enhanced or novel properties. While microbial enzymes have favorable turnover rates, They often need fine tuning to meet industrial production requirements. Challenges such as substrate oblique product inhibition, stability, narrow substrate specificity and enantiomer selectivity need to be addressed. Genetic modification, particularly recombinant DNA techniques, has significantly boosted enzyme production, increasing yields up to 100-fold. Developing new or improved biocatalyst is a task with two main approaches. The first one is the rational design of existing enzymes to enhance their properties, and second one is combinatorial methods, which involves creating random libraries to identify enzymes with the desired functionality. Let us start with the rational design. This approach involves site directed mutagenesis to target specific amino acid substitutions requiring detailed knowledge of the enzyme's 3D structure and reaction mechanism, which may not always be available. However, the growth of protein structure and sequence databases is helping to address this limitation. By comparing new biocatalysts with known sequences in these databases, researchers can identify related proteins with known functions and structures. Since new enzymes evolve through minor modifications to active site structures, homology driven experiments focus on engineering binding sites for different substrates and creating new catalytic residues to modify enzyme functions. Site directed mutagenesis is a powerful technique which is used to modify genes and investigate the structural and functional properties of proteins, particularly focusing on enzyme structure, function, catalytic mechanisms and residues. Site directed mutagenesis involves both single and combinatorial mutations and typically analyzed using bioinformatics tools. By combining site directed mutagenesis with other techniques, enzyme properties can be significantly enhanced. For example, the yield of maltose binding protein fused HepI from recombinant E. coli was improved by 30.6% through a strategy that combined site directed mutagenesis with the addition of calcium ions for thermostabilization. Let us now look how site directed mutagenesis is introduced. Target genes are identified and specific nucleotides are selected to be modified, creating synthetic primers that include the desired mutation. So, here we have a forward primer which is carrying the desired mutation or the changed nucleotide at this particular position, as shown by this red asterisk. The gene is amplified using these particular primer by the PCR method, polymerase chain reaction method, ensuring the mutation is incorporated into the new DNA strands that is synthesized by this polymerase chain reaction. Then the mutated DNA is introduced into host cells to propagate and express the gene. So, here we have parallelly also carried out a DNA replication with a reverse primer which carries the complementary mutation, and once both the strands are synthesized, these are annealed and put together. So, these particular construct are now then used for transformation into the host, and then we finally go for screening of the desired mutants. So, transform cells are screened to identify those containing the correct mutation, and we will go for DNA sequencing to confirm the accuracy. Then mutant protein is expressed and its properties like activity, stability, structure are analyzed. The site directed saturation mutagenesis is an advanced technique for the rapid evolution of proteins, where each amino acid in a protein is systematically replaced with all other 19 naturally occurring amino acids. This process focuses on hotspot regions of enzymes where mutations can lead to improvements in properties like thermostability or catalytic efficiency. A newer approach called combinatorial co-evolving site saturation mutagenesis was introduced by Wang et al. In 2012. In this method, functionally Correlated variation sites are targeted for mutation creating libraries of mutants. This technique has been shown to enhance the thermostability of alpha amylase from Bacillus subtilis CN7 by 8 degrees centigrade, demonstrating its potential for improving protein properties in a directed and efficient manner. Let us now discuss about directed evolution. This enables the creation of large libraries of enzyme variants that can be screened for desirable properties such as enantioselectivity, catalytic efficiency, solubility specificity and stability. This approach does not require any prior knowledge of the enzyme's structure or function, making it a fast and cost effective technique for improving enzymes. Directed evolution mimics natural evolutionary processes by introducing genetic diversity through random mutagenesis techniques. After mutagenesis, the enzyme variants are cloned, expressed and then screened based on their performance under specific conditions. So, this is one of the most important technique. So, here we have a gene for example, where we go for mutagenic PCR and create a gene library or pool. So, these are all cloned and expressed in a host like E. coli, and then we go for activity assay, and the clones which are not having any activity or desired activity are rejected, and the clones which are having the desired variants are isolated, and then these can be moved into the next cycle. And so, with every cycle there is a process of evolution taking place, but this evolution is not random, it is a directed evolution because we are using mutagenic PCR to influence these evolutionary process. So, that is why this is mimicking the natural evolutionary process by introducing genetic diversity through random mutagenesis techniques and selecting the desired variants at every cycle and subjecting them to further evolutionary pressure. So, the mutagenesis techniques that are used in directed evolution for introduction of variation includes error prone PCR, and this introduces random point mutations in the gene encoding the enzyme, generating a diverse pool of variants. Then oligonucleotide directed mutagenesis, this uses specific primers to introduce mutations at predefined sites. Then we may also go for chemical mutagenesis, where we employ chemical agents to introduce mutations in the DNA. And then there are several other techniques like DNA shuffling or molecular breeding. Then these facilitate random homologous recombination between parent genes, typically with over 70% homology to create a new pool of gene variants. Let us now discuss about the recombinant protein expression. Advances in recombinant DNA technology have enabled efficient expression systems for producing enzymes from various microorganisms using industrial hosts such as E. coli, Bacillus subtilis, Ralstonia eutropha, Pseudomonas fluorescens, Saccharomyces cerevisiae, Pichia pastoris and Aspergillus species. Use of recombinant rDNA technology has several advantages. Number one, higher yield and productivity. The overexpression boosts enzyme production efficiency, then improves stability and activity, enhanced thermostability, pH tolerance and efficiency. Control and consistent production ensures reliable batch to batch enzyme quality, customized enzyme functionality tailored for specific industrial needs and resistance. Cost effectiveness lowers production cost by optimizing microbial growth and yields. Notably, around 90% of industrial enzymes are recombinants. What are the steps in the recombinant DNA technology? So, this we are also discussing in other sections. So, let us just have a quick overview. So, here is a plasmid vector. Plasmids are extra chromosomal DNA, as you all know, which helps the host overcome stress conditions, but we also use those plasmids for transferring foreign genes. Then from another source we isolate the gene of interest, and then we do restriction endonuclease digestion of both the plasmid vector and the gene cut out from the genomic DNA, and we ligate them. And these construct is then used for transforming into the host, and we screen the cells which are thereby transformed. So, this is the general overall scheme. So, isolation of the vector gene vector, isolation of the target gene, then cleavage of isolated DNA particular site by restriction enzyme, ligation of the digested target gene with the vector DNA, introduction of rDNA into compatible host cells, replication and expression of rDNA within the host cells, screening of the transformed cells. So, expression of enzymes in E. coli by this cloning method. E. coli is one of the most extensively used recombinant host for protein production due to several advantages. Number one, it is very easy to manipulate it genetically. It grows very fast and it is cost effective when it comes to its culture and maintenance. And then it has very low protease activity and it has high protein accumulation. The low protease activity helps us in obtaining more proteins. Otherwise, if the protease activity is high, that will actually degrade the proteins that are produced by this process. However, despite its advantages, E. coli has some significant drawbacks such as its lack of post-translational modification and formation of inclusion bodies. Despite these limitations, E. coli has been used successfully to achieve high level production and secretion of certain heterologous proteins such as alkaline phosphatases and Levan fructotransferases. Let us discuss about the strategies of high level expression in E. coli. High level expression by translational modification. High level protein synthesis in prokaryotes requires strong promoters and precise regulation to control gene expression. Thermal or chemical inducers are commonly used to activate these promoters. Efficient transcription terminators are also essential for enhancing mRNA stability and boosting protein yields. Anti-termination elements such as the transcriptional anti-termination regions from the E. coli, rRNB, rRNA operon have been incorporated into the expression systems like the PAC420 vector to improve heterologous gene expression. High level expression by translational modification, the secondary structure of the mRNA at the translation initiation region and the overall stability of mRNA significantly impacts gene expression efficiency. Then we also can increase the level of expression by codon optimization. Heterologous protein production in E. coli can be impacted by biased codon usage as the organism shows a non-random preference for synonymous codons. Rare tRNAs can compete during translation, leading to issues such as mistranslation or lower protein expression, especially with rare codons like AGA and AGG for arginine. Codon optimization can improve expression by altering rare codons to more common ones or by co-expressing tRNA genes to exist in translation. Using synthetic DNA to create codon optimization genes without secondary structure issues has proven successful in enhancing protein production. So, some of the other expression systems other than E. coli include Saccharomyces cerevisiae. These Saccharomyces cerevisiae offer several benefits as a cloning host, which includes rapid growth in simple media, then high cell density and the ability to secrete proteins. It has been successfully used to produce glucose oxidase from Aspergillus niger at 9 gram per liter. Then we have the Picia pastoris, which has become a popular expression system with over 700 proteins produced, including recombinant proteins reaching up to 22 gram per liter compared to 9 gram per liter in Saccharomyces cerevisiae intracellularly, and 14.8 gram per liter in the secreted form, which is again higher than that produced by Aspergillus niger at 9 gram per liter. These advantages of Picia pastoris over Saccharomyces cerevisiae therefore include efficient and tightly regulated methanol promoters, reduced glycosylation, high secretion levels of proteins, and easy scale up for high density growth. Then we have another expression system, the Hansonella polymorpha. Heterologous gene expression in Hansulella polymorpha is similar to Picia pastoris, utilizing the methanol oxidase gene promoter for foreign gene expression. It can produce around 1.4 gram per liter of secreted glucoamylase and 13.5 gram per liter of phytase. Filamentous fungi are also used as an expression system. For example, molds like Aspergillus awamori produces around 4.6 gram per liter of glucoamylases, then Aspergillus oryzae 3.3 gram per liter of mucorrenin, Acremonium chrysogenum 4 gram per liter of fusarium alkaline protease, and Trichodoma raceyi 35 gram per liter of recombinant proteins, which is quite impressive. And these have had various success in recombinant protein production. Chrysoporium lucknowense is a recent entrant producing around 50 to 80 gram per liter of proteins with low viscosity and low protease mutants. Some enzyme protein domains are not essential for their activity, and thus random or directed truncation methods to remove them such as site directed truncation and random truncation can be used to modify these enzyme properties. So, truncation is an important step by which we can enhance the properties of certain enzymes. These involves removing amino acid sequences that are not essential, such that only the main catalytic domain remains. By truncating parts of the enzyme, a truncation library can be generated, leading to variants with altered or improved activities. For example, after truncation, an endodextranase mutant TMNCG delta, this delta stands for the truncation or the deletion, from Streptococcus mutant ATCC 25175 exhibited hydrolytic activity on 0.4% dextran. T2000 similar to the wild type enzyme SMDEX90. Additionally, it demonstrated 1.4 fold and 2 fold increased activity on 0.05% dextran T2000 and T10 respectively. Another technique is that of creating fusion proteins, so which are also called as chimeric proteins or chimeric enzymes. These are basically engineered by fusing catalytic and substrate binding domains from different enzymes, and it has demonstrated enhanced properties such as improved thermostability, catalytic activity, substrate specificity, and product selectivity. So, here in this illustration we can see in figure A general scheme for creating chimeric cellulose degrading enzyme by fusing the carbohydrate binding domain from organism A with a catalytic domain of organism B in bottom for digestion of cellulose by the chimeric enzyme. So, here this is the gene for carbohydrate binding domain, and here is the gene for catalytic domain. So, these are fused to create a fusion DNA, and these are transformed and expressed. So, you have now a fusion protein where you have the carbohydrate binding domain from one source and the catalytic domain from another source linked to one another by a linker in between. And then in figure B, this fusion protein or chimeric protein enzyme is used for degrading cellulose. Functional genes or oligopeptides can be incorporated to create chimeric enzymes with unique features. So, here already we discussed about these catalytic module and the carbohydrate binding module connected by this flexible linker. CBMs classified into 54 families based on amino acid similarity and ligand specificity are commonly fused with other enzymes to create chimeric enzymes with enhanced or multiple activities. Let us now discuss about applications of microbial enzymes in various industries, particularly their application in organic synthesis industries, in therapeutics or pharmaceuticals, in diagnostics, in food and beverage industries, in animal feed production, in paper and pulp industries, in textile industries, in leather industries, in detergent industries, and in waste management. So, these tables show some of applications of enzymes in various industries. For example, in food and beverage industries, we use proteases, lipases, lactases, pectinases, amylases, lacases, amyloglucosidases, phospholipases for the degradation of starch and proteins into sugars, production of low calorific beer, fruit juice processing, cheese production, glucose production from lactose, DAO stability and conditioning. Similarly, in the detergent industry, we use amylase, cellulase, lipase, protease, manases for removing proteins after staining, cleaning agents, removing insoluble starch, fats and oils to increase effectiveness of detergents. Then in the textile industry, we use amylase, cellulase, pectinase, catalase, protease, peroxidase, keratinase for giving finishing to fabrics in denims, wool treatment, degumming of raw silk or biopolishing, cotton softening. And in the animal feed industry, we use phytase, geilanase for increasing total phosphorus content for growth and digestibility. In the ethanol production industry, we use cellulases, ligninase, manganese for the formation of ethanol. In paper and pulp industry, we use amylase, lipase, protease, cellulase, hemicellulose, esterase, ligninase, geilanase for degrading starch to lower viscosity, aiding sizing, de-inking and coating paper, cellulose and hemicellulose smooth fibers, enhance water drainage and promote ink removal. Lipases reduce pitch and ligninase remove lignin to soften paper. Then in the leather industry, we use proteases and lipases, unhairing, batting and depicking. And in the pharmaceuticals, we use penicillin, acylase, peroxidase for the synthesis of semi-synthetic antibiotics and production of antimicrobials. In the molecular biology, we use DNA ligase, restriction enzymes, polymerases to manipulate DNA in genetic engineering, DNA restriction and the polymerase chain reaction, and also important in forensic sciences. Now, let us discuss the application of microbial enzymes in organic synthesis. Enzyme based processes are increasingly important in fine chemical production due to their ability to deliver high purity products in an eco-friendly and cost effective manner. Microbial catalyst used for centuries in alcohol and cheese production now plays a key role in industrial synthetic chemistry. Lipases are among the most commonly used enzymes, particularly for producing optically active alcohols, acids, esters and lactones such as intermediates for diltiazem. Oxidoreductases like polyphenol oxidase are essential in synthesizing DOPA, a treatment for Parkinson's disease. Then the glycosyl transferases, which are employed for the regio and stereoselective synthesis of oligosaccharides and polysaccharides, crucial for cellular recognition and communication. Other enzymes such as lyases are used to synthesize compounds like cyanohydrins, acrylamide and malic acid. Now, let us discuss about the use of microbial enzymes in pharmaceuticals or therapeutics. Enzymes play crucial roles in the pharmaceutical and diagnostic industries, serving as the therapeutic agents for conditions such as enzymatic deficiencies and digestive disorders, and are essential tools in diagnostic procedures like ELISA and diabetes testing kits. We will be discussing about the diagnostics separately. So, the medical applications of microbial enzymes are diverse and rapidly expanding, including enzymatic deficiencies, digestive disorders and clot related issues. They are also used in specialized treatments such as producing intermediates for medications, synthesizing therapeutic compounds and generating bioactive molecules with antibacterial, antioxidants and anti-tumor properties. Some of the major enzymes are listed in the table on slide 52 along with their functions. So, here we have enzymes like L-asparagine, L-glutaminase, L-tyrosinase, galactosidase, which are used for treating tumors, and the microorganisms which are used for producing these are the E. coli, Pseudomonas, Acidovirans, Beauveria bassiana and Acinetobacter. Then superoxidase, dismutase and serrapeptase are anti-inflammatory agents produced in Lactobacillus, Nocardia and Mycobacterium. Streptokinase, urokinase acts as anticoagulants produced in Streptococci, Bacillus and then penicillin oxidase, rifamycin B oxidase. These are used for antibiotic synthesis producing penicillin, and superoxide dismutase glutathione peroxidase catalase acts as antioxidants, and these are produced in Lactobacillus plantarum and C. Glutamicum. Then we have collagenase, which are used for treating skin ulcers, produced in Clostridium perfringens. Then we have lacase, rhodamase. These are used as detoxification agents, and they are produced in Pseudomonas aeruginosa. Then we have beta lactamase, which are associated with antibiotic resistance, produced in Klebsiella pneumonia and Citrobacter frundi or Serratia marcescens. Then we have the uricase, which are treatment for gout, produced in Aspergillus flavus. Similarly, we have alpha amylase and lipase used in digestive disorders, produced in Bacillus species, etc., and rhodamase, which is used in cyanide poisoning treatment and produced in Sulfobacillus sibiricus. Let us now discuss about the application of microbial enzymes in the diagnostic area. These enzymes enable the measurement of biomarkers for various health conditions. So, some of the examples include glucose oxidase for glucose, urease and glutamate dehydrogenase for urea, and urate oxidase for uric acid. Then we have cholesterol oxidase, detects and converts cholesterol, while putrescine oxidase identifies biogenic amines such as putrescine, a food spoilage marker. In genetically engineered diagnostic tools, enzymes are vital for nucleic acid manipulation. Restriction endonucleases facilitate site specific DNA cleavage for molecular cloning, and DNA polymerases enable DNA amplification through polymerase chain reaction. And then comes the application of microbial enzymes in the food and beverage industry. The understanding of enzymes roles in the food manufacturing and ingredients industry has enhanced processes leading to safer, higher quality products. Enzymes have expanded into new areas like fat modification and sweetener technology. Enzymes in this industry are used to improve brewing consistency, enhance the nutritional properties of proteins, and increase juicy yield while improving color and aroma. The applications span baking, dairy, juice production, and brewing. In baking, microbial enzymes improve dough stability, crumb texture, and product shelf life. In dairy, enzymes are essential for cheese processing, making it the second largest application area, followed by the beverages. Microbial enzymes are also used in the production of animal feed. Feed enzymes are used in animal diets to break down harmful or non-nutritive feed components and enhance the nutritional value, particularly for poultry. Common feed enzymes include phytases, proteases, alpha galactosidases, glucanases, gyanases, alpha amylases, and polygalactouraneases. Phytase is the largest enzyme segment in the feed industry, which enables the utilization of phosphorus bound in phytic acid in cereal based feed, thereby improving phosphorus availability. Monogastric animals are unable to digest cellulose and hemicellulose in plant based feeds, and they benefit from gyalanase and beta glucanases, which aid in the starch digestion. Proteases break down proteins into amino acids, reducing anti-nutritional factors and enhancing feed efficiency. Microbial enzymes are also used in the paper and pulp industry. With growing awareness of sustainability, the use of microbial enzymes in the paper and pulp industry is increasing in order to mitigate environmental impact by other conventional procedure. Enzymes reduce the processing time, energy consumption, and chemical uses, while enhancing processes like de-inking, bleaching, and waste treatment by improving biological and chemical oxygen demand. Gyanases and ligninases remove lignin and hemocellulases, improving pulp quality. Amylases are used for starch coating, de-inking, paper cleanliness, and drainage improvement. Lipases aid in pitch control and de-inking, while cellulases are applied for de-inking, softening and drainage enhancement, as well as recycling printed paper. Lacases serve as a chlorine free alternative in chemical pulping. thereby reducing ozone depleting and acidifying waste. Mannanases improve paper brightness by degrading glucomannan. These enzyme applications make paper production more sustainable and eco-friendly. Microbial enzymes are also used in the leather industry. These leather industry faces significant environmental and health challenges due to the discharge of waste and harmful chemicals like sulfides, lime and amines during various processing stages. Enzymes offer a biodegradable and eco-friendly alternative, improving leather quality while reducing waste and pollution. Key enzymes like proteases, alkaline and neutral lipases and amylases are used across processes such as dehairing, soaking, batting, degreasing and tanning. Additionally, enzymatic dehairing preserves the hair, reduces reliance on harmful chemicals and produces softer, more pliable leather. So, here we can see the environment of a leather tanning industry. Enzymes are now being used for more eco-friendly and sustainable tanning process. Similarly, microbial enzymes are also used in the textile industry, which normally generates significant waste from processes like fabric desizing, bleaching and dyeing, making it a major contributor to environmental pollution. Enzymes are increasingly used to develop eco-friendly technologies of fiber processing while enhancing product quality. Hydrolysis such as amylase, cellulase, cutinase, protease, pectinase and lipase are used for processes like biopolishing, biosourcing, anti-felting of wool, cotton softening, denim finishing, desizing and synthetic fiber modification. Then there are the oxidoreductases, which include catalase, lacase, peroxidase and ligninase, which are employed for bio-bleaching, bleach termination, dye decolorization and fabric finishing. So, in this table we can see some applications of enzymes in the textile industry. For example, amylase is used for desizing, it is produced in Bacillus. Cellulose used for cotton softening, denim finishing produced by Aspergillus penicillium. Then we have catalase, which is used for bleach termination produced by Aspergillus. Lacases is a non-chlorine bleaching fabric dyeing produced by Bacillus. Then pectate lyase bioscouring, so produced by Bacillus. Then comes protease, which is used for removal of wool fiber scales or degumming of silks produced by Aspergillus niger. Then we have the lipases, which is used for removal of size lubricants and denim finishing produced by Candida antarctica. Then we have ligninase used for wool finishing produced by Trematis, and then collagenase which is also used for wool finishing and produced by Clostridium histolyticum. And then we have cutinase which is used for cotton scouring, synthetic fiber modification produced by Pseudomonas etc. Now, let us discuss about the microbial enzymes which are used in the detergent industry. These enzymes play a key role in the development of industrial detergents widely used to remove protein, starch, oil and fat based stains, and they include amylase, lipase, cellulase, cutinase and proteases. Cellulases brighten colors, soften fabrics and remove small fibers without harming major ones, while cutinases is used for lipolytic cleaning in dishwashing and laundry detergents. In dishwashing detergents, proteases and amylases effectively remove protein and carbohydrate based food residues. Microbial enzymes are also used for waste management. They play a vital role facilitating the degradation of toxic pollutants like phenols, aromatic amines and nitriles from industrial effluents and domestic wastes. They are used individually or in combinations, convert harmful compounds into harmless products through degradation or bioconversion processes. A wide range of enzymes is employed in waste treatment, including amylases, celluloses, lipases, nitrile hydratases, pectinases and proteases. Oxidoreductases such as lacase, manganese peroxidase, lignin peroxidase and tyrosinase are particularly effective in detoxifying organic compounds through oxidative coupling. They remove chlorinated phenolic compounds from industrial effluents. Then we have the oxygenases, including monooxygenases and dioxygenases derived from microbial source. They exhibit broad substrate specificity and are instrumental in degrading halogenated organic pollutants like herbicides, insecticides, fungicides and industrial fluids. So, with this we come to end of today's lecture. Thank you for your kind attention.